Hello, good afternoon. Hope you guys had a good weekend. We are going to be taking up the test one review today, and you can find a review on eCentennial. If you don't already have a copy, that's okay. Just try to follow along with the questions, and I'll post solutions afterwards. All right. Now, this is not our comprehensive review. Okay. Like, I mean, it has a lot of information on it, but then I couldn't fit everything onto the review. So just because the topics are not covered on the review does not mean it's not going to be on the test. Okay. So please make sure you can start with the review package, but then you also should study a little bit more, especially things that were not included uh, on the list. Okay, let me just try to copy them over, and then we're gonna get going. Okay, so here are a few questions about the beetroot experiment. Um, First question, what is the control of the experiment? Okay, so the control is the variable that um, you are going to be comparing with. It's the group that has not received any treatment, no changes. It provides a reference and a baseline to compare all the other stuff with. And in the beetroot experiment, that would be the test tube um, that is at room temperature. Okay, so the room temperature test tube or beaker, right? Uh, Next, what is the dependent and independent variable? Okay, so the dependent variable is the one that you'll be measuring or observing. In the case of the beetroot experiment, that would be the membrane permeability. And we actually assessed the membrane permeability using two separate ways. We did it qualitatively, which is just based on the color intensity. Okay, you're just looking at the color difference. You're not doing any kind of numeric measurements, and then you measure it quantitatively. Sorry, quantitatively, which means we are using um, the colorimeter, right, to measure green light absorbance. Okay. Sorry, I think I spelled quantitative wrong here. Let me try that again. Quantitative. There we go. Okay, so qualitatively is just using your senses. Quantitatively is to use measurements, numbers. Right? So these are the dependent variable, and the independent variable would be temperature. That's it. Okay, what variables are standardized? Standardizing variables allow us to be certain that any differences we see in the membrane permeability is actually caused by the temperature difference and not anything else. Right? So an example of that would be the size, the size of the beetroot discs. So when we cut the beetroot into little cylinders, we have to make sure that they're all the same size. If some are big and some are small, the bigger one might release more pigment, right? Then we cannot say for sure it was due to the temperature, and instead of like the size difference. Does anybody have an other standardized variable? Do you remember anything that you might put for the lab?
The quantity amount of the water or something.
That's right, yeah. So the volume, right? Volume of water in the test tube, right? If you put too much or too like m more in one test tube and less in the other, then you will dilute the color, right? And so the volume of the water is going to be important. Duration of, you can just say, time spent in the beaker. Right? You want them to all sit there for 1 minute at the respective temperature, right? So not some are going to be longer or some are going to be shorter. That's obviously going to affect your permeability. So you want them to be the same time. Okay, so these are some standardized variables. All right, next question. Why does a frozen beetroot have similar color intensity as 80 degrees when placed in to water at room temperature? Okay, so this question was something we talked about in lecture four. It was part B of lecture four. I told you at the time when you freeze something, when you freeze a cell, the ice crystal basically puncture or damages the cell membrane. Okay, so when you freeze it, the cell membrane of the plant is already full of holes because of the ice. So when you thaw it, it doesn't really matter that it wasn't heated before because it was already full of holes. So all the pigment is gonna leak out. Right? So that's why you're gonna see a very intense color, um, so intense that it's similar to 80 degrees. Okay, what do you expect the absorbance to be if we included temperatures higher than 80 degrees in our experiment? So if you plot the graph, this is absorbance, percent absorbance, and then this is temperature. Your graph looks something like this, right? Okay, like it's slow increase and then it kind of flat out here. This actually happened at around 70 degrees. Okay, many of you said that it peaked at 80 degrees, but the question asked you like, when does it like stop having drastic changes, right? I still give you mark if you say 80, okay, but then like the correct answer should be like around 70. And what happens is, by the time you get at 70 degrees, basically the cell membrane is completely destroyed. Okay, so like heating it up more is not going to destroy it further. Right? It's as destroyed as it can be. So you wouldn't expect the absorbance to go up any further, right? You would expect it to be similar absorbance as 80 degrees. Okay? Like, can it be like 1 or 2% higher? Sure, but that's not because there are more pigment leaking out, right? It's just like subtle fluctuations, right? It's not, certainly it's not going to go beyond 100%. Many students put like it's going to go to 120%, 140%. It's not possible. Absorbance, 100% is absolute highest. You cannot absorb more than what you are shining at the test tube, right? So that's the answer. Does anybody have any questions about the beetroot stuff before I move on to lecture one?
Oh, yeah. So basically, we have our grades, but is it possible for us to have the work back at some?
Yeah, like I said from before, right, like we don't really return stuff for the online assessment. I'm really sorry about that.
Okay.
Uh, but for the tests, these are the only thing I'll test you on regarding the lab. Okay?
Okay.
So
All
if you
right.
understand these five questions, you should be okay for the lab portion of the questions. Okay? Amberpreet, please go ahead.
What is the reason for the measuring green light absorbance with the calorimeter?
Yeah, sure, right. So like the color we are trying to observe here is a red color. Okay. So if you think about the color wheel, um, red is going to reflect red light, okay? And that means it's going to absorb everything else. So if you shine a green light at it, depending on how red the solution is, it's going to either absorb more green light or less green light. The more intense the red, the more green light it is going to absorb. So basically, the machine, the colorimeter is going to shine green light at it, and then it's going to measure how much green light passes through, and then it does some calculation, and it tells you, give you a number, give you absorbance. Higher absorbance means more intense the color is. It's just like a numeric way to compare the colors of the different test tubes. Okay. Green is easier to get because it's lower energy compared to something like blue. Blue is closer to like purple, right? Like we know the sun has UV light, ultraviolet, right? Which is like the blue end, purple end. Those are higher energy. So green is easier, more accessible. Does that answer your question?
Yes, thank you.
Thank you. Okay, that's it for the beetroot. Feel free to let me know if you have more question about it. Next, we're gonna do the lecture one, scientific method. I'm just gonna copy first two questions here. Okay, what is a placebo? So placebo, the best way to think about it is that it is a sham treatment. Okay, sham treatment means it has no actual effect. No actual effects on the dependent variable. Right? It's like a sugar pill. And we usually need a placebo to minimize bias and psychological effect, right? Can't spell today, sorry. Ecological effects. Right? So give an example of an experiment where a placebo is needed. So in class, we say, we need a placebo when we involve human subjects. So e.g. testing a new sleeping pill. Okay? We're going to need a placebo for that. Give an example where placebo is not needed. Okay, so in class, we talk about testing cows, um, different diet, and milk yield, right? Animals not gonna have bias, so we don't really need a placebo. Okay. Now, in some cases, the word placebo and control might be used interchangeably. It depends on the context. Okay. But for us, just keep it simple. Okay. Like when you involve humans in a test in an experiment, you're gonna need a placebo. Otherwise, you don't need a placebo, but you still need to have control though, right? So that's that. What is a valid hypothesis? Okay. So as you may recall, Valid hypothesis must be number one, testable, and number two, falsifiable. Okay? You need to be able to run an experiment to test it, and it must be potentially wrong. Okay? And we can practice writing hypothesis. For example, in the beetroot lab, a good hypothesis would be higher temperature will increase the membrane permeability, resulting in more pigment being released into the water. Okay. Many people say higher temperature will affect membrane permeability, but without saying how. Like, is it going to increase it? Is it going to decrease it? Like, you don't have to explain like why it is affecting the membrane permeability, not in the hypothesis anyways, but you have to state the relationship, right? Like, is it going to make it more permeable, less permeable? And then, we attach a little bit of a prediction with our hypothesis here. Okay, so that's that. Hey, let me copy number 3 over. All right, so here we have the sleeping pill. We talk about this in class, right, lecture, but let's go through it again. We have control, which is also known as the placebo. Again, like I said, it's interchangeable in the context of doing experiment with humans. These people are basically receiving a sugar pill. Okay? They don't know about it, but we're giving them something. Okay? So here, the sleeping pill is supposed to reduce the symptoms of jet lag. And on the left graph here, we can see there is a reduction in jet lag symptoms in both the placebo and the experimental drug. So based on this result, do you think the drug is effective? Anybody? If I show you this graph on the test and I ask you, is the drug effective or not in reducing the symptoms of jet lag? What are you going to say?
Not effective.
Not effective. That's right. How come? This is ineffective. I agree. But why?
There's no change in symptoms.
But there is change though. Like, it's less people, right? Like, we only have 40% of the people, less than 40% of the people experiencing jet lag.
People that are psychologically believe that it helps to reduce the jet lag.
Okay, yeah, basically, right? Like, I think you're on the right track here. The key thing to articulate here is that the control and the treatment both were able to reduce the symptoms of the jet lag. In other words, eating a piece of candy is going to help you reduce jet lag just as good as the medication. So why would you take the medication? You might as well just take the candy, right? The sugar pill, right? Okay, so it's ineffective because both placebo and the drug were able to reduce symptoms. Okay, suggesting this is more of a psychological effect. Okay. So it looks like they could take anything. As long as you tell them it's going to help them sleep, they're going to feel better about it. And then therefore they report less symptoms, right? So on the other hand, on the right side here, this would be what effective looks like. Effective. Only the drug has significant symptom reduction. Okay. So like taking the placebo. Remember, placebo is not supposed to do anything, right? That's why we see the people not um, relieving their symptoms, right? A high percent of people still report having symptoms. Like you might argue, hey, how come like, I see like a 20% reduction here? Doesn't that count? Well, some people just are not bothered by jet lag in general, right? So that's probably what that 20% is. But you see a significant more reduction in treatment compared to the placebo. That's how you know it's effective. Okay, other things you should remember to study from lecture 1 would be here, let's see. Don't forget there are the experiments, right, with Rady and Pasteur. Okay, I don't have time to review those with you, but you should on your own. Characteristics of living things. Okay. And then primary and secondary sources. And so those are some of the other stuff that we learn in lecture 1. Any questions from lecture 1 before I move on to lecture 2? Okay. Like I said, you'll have more time this time for the quiz, for the test. Okay, and there are no written response, so it should be a little bit better. Okay, lecture 2. What holds the atoms together in a water molecule? Okay, so water molecule is, of course, as you know by now, it's H2O. Okay, so you can draw a water molecule. There it is. Okay. Now, it's asking what holds the atoms together. This here, the bond that holds the oxygen and the hydrogen together, it's called covalent bond, which is sharing of electron, as opposed to like transfer of electron. But in this case, for the water, the sharing of electron is actually uneven. So the oxygen ends up slightly negative compared to the hydrogen. So this is slightly negative. I'm just gonna write negative, I guess. And the hydrogen atoms are slightly positive. Excuse me. So if you put two water molecules next to each other, like that, the slightly positive portion will be attracted to the slightly negative portion of another molecule, and this interaction is called hydrogen bond. Okay. So hydrogen bond also exists in secondary and tertiary protein structure. And also exist in DNA. It holds bases of complementary strands together. Okay. I know this question is only asking about water, but I'm just like adding things here and there that relates to other concepts that you should know. Okay, like I'm not adding things that you don't need to know for the test. These are all things that we've learned and things that you should remember for the test. Okay, next. If you supply radioactive nitrogen atoms, which is something that you can trace it, okay, and detect it. If you supply that to a cell and let it grow, what molecules will you expect to find the radioactive labels in? So it sounds like a, strange question, but basically it's just asking you what kind of molecules in the cell has nitrogen. Okay, so we of course have four kind of macromolecules. So carbohydrates, proteins, nucleic acid, and lipids. Okay, so let's think about the atoms that build each of these things. Starting with carbohydrates, what are the key atoms that you will find in carbohydrates?
Carbon, hydrogen, oxygen.
Yes, carbon, hydrogen, and oxygen, right? In fact, all of them has carbon, hydrogen, and oxygen, right? And that's it for carbohydrates. For proteins, in addition to carbon, hydrogen, and oxygen, there are also nitrogen and trace of what?
Sulfur?
Phosphorus.
Sulfur,
Oh,
yeah, sulfur, yeah, that's okay.
yeah.
Sulfur is good. Nucleic acid, we have N and P, and then
Sulfur.
that's right. This is the one that has trace of phosphorus.
No, phosphorus lipids.
Yeah, that's right for lipids, particularly the phospholipids, right? So the question is asking, hey, where will you find the radioactive nitrogen? Well, it's going to be in the proteins and the nucleic acid, right? That's what the question is asking. Hey, next. Regarding DNA, what is the monomer for DNA? So there are a couple things going on here. I'm just going to answer all of them here. So first of all, the DNA is made up of nucleotides. Okay. This is the monomer. Now, for DNA specifically, as opposed to DNA and RNA, for DNA specifically, the nucleotide is going to have the phosphate, okay, PO4, -3, is going to connect to a sugar, and this sugar is called deoxyribose sugar. It's missing an oxygen compared to RNA, and it's going to be connected to a nitrogenous base. For DNA, this base, this nitrogenous base, can be one of four things. And you should know this by now. I'm sure you do. A, C, G, and T. So that's a nucleotide. You connect a bunch of these things together, you get your DNA strand, right? How are nitrogenous base classified? Well, nitrogenous base can be purines, which is 2 rings, and that includes A and G. And remember, I told you to think of the phrase pure as gold. That's how you know it's A and G. And there are pyrimidines, which is only 1 ring, and that includes T and C. As for the base pairing rule, adenine pairs with thymine. And then there are 2 hydrogen bonds that would be holding them together. Whereas C and G, there will be 3 hydrogen bonds holding them together. Okay, so it's going to take more energy to separate DNA that has a higher CG content compared to AT contents. Okay, so what force holds the nitrogen base together? Hydrogen bond. Okay, so that's D. What is the significance of base pairing rule? Well, the significance is they are complementary. So knowing just one side of the DNA is enough to build the entire strand, the entire double helix. Okay, knowing one side of the ladder will allow you to build the missing half. Right? It turns out this is how DNA are replicated. We'll learn that in the second half of the course. That's DNA right there. Okay. Let's take a look at the next question. Oops. What is dehydration synthesis? Illustrate with a diagram the significance of this process. Okay, so I'm gonna review a couple things here at the same time. Hopefully it's gonna make sense. So we have the individual building blocks. Okay, so these are monomers. And what we can do is we can combine them together. We can join them together to form a long chain. Okay, so this long chain is the polymer. Okay, monomers and polymer. And the process of connecting them together, the name of the process is dehydration synthesis. Okay, also known as condensation reaction. Okay, and the reason it's called dehydration synthesis is because you actually need to remove H2O. So that's it, right? Now I'm going to add a few more things here to review with you. We can actually break the polymer back into monomer, but in doing so, we're going to need to add water. Okay, we'll say this is remove water. So this is a hydrolysis. Hydrolysis. and it will allow us to break it back down into monomers, right? Now, also important in this situation is that when we break down the polymer, it actually releases energy. Energy. That energy is captured by ADP and phosphate to generate ATP. Okay? And we can use that energy to help build things in the body. So you might also remember when we break things down, that's catabolism in the body. Cellular respiration, it's a catabolic reaction. And here, when you build molecules that you need, that's anabolism. Okay, so everything is kind of connected in the course. Any questions? All right. Next, regarding enzyme. What is an enzyme? How does it work? Factors that affects the functionality of an enzyme. Okay, so let's review all that together. First of all, an enzyme is it's a protein, right? It's a protein that catalyzes or speeds up reaction in the body. Okay. And this is an example we recently did. Okay, so this is an enzyme. And the pocket that we fit the ingredients in, those are called the active site. Okay, where the substrate fits. And this, I'm just gonna use substrates because there are more than one. Substrates are the ingredient, right? So recently we learned that there could be a donor molecule with a phosphate on it, and then with the help of an enzyme, you can transfer it onto a molecule of ADP. I hope this sounds familiar to you. Okay. So these here, these ingredients, those are the substrate, right? So actually, anybody remember what this process is called? Like not even the full name, just like the acronyms for it, the abbreviation for it. What is it? Oh, well, this is how we produce ATP, yes, my cup. But what's the name of the process?
Substrate level phosphorylation.
Yeah, that's the one, right? Substrate level phosphorylation, the SLP, right? Okay, so that involves an enzyme. Okay, so how does it work? Well, enzyme works by lowering the activation energy. Activation energy is the energy we need to supply before a reaction can take place. Usually that amount of energy is quite high and it's not sustainable in the body. So with the help of an enzyme, you reduce that activation energy. What factors affect the functionality of an enzyme? Well, the enzyme is a protein, so anything that affects the structure, the shape of the protein is going to have an effect on the enzyme. Things that we talked about in class include temperature, pH, salt concentration, right? All these things would affect the shape of the protein. If you change the shape of the protein, if you change the shape of the enzyme, the pocket might be of a different shape. And if you have a different pocket, different active site, then you will no longer be able to fit the substrate in. Okay, lastly, what does the enzyme lactase do? Lactase, if it ends with ASE, 9 out of 10 times it's an enzyme. So this is lactose. And then we put the enzyme on top of the reaction arrow, something that you probably already learned in chemistry. And the lactose is going to be broken down into glucose plus galactose. So that's the enzyme. This is the substrate. Okay? These are the products. Substrates are the ingredient. Okay? We only call them substrate if we have an enzyme. Okay? Otherwise, they're just called reactants. Okay, that's that. Okay, next. Here is the table. It's very long. So I'm going to try to fill it in with you guys. Okay. I'm just going to do the function and the examples together. That usually works a little bit better. Okay, monomers. For carbohydrates, the generic name for all the monomers are monosaccharide. The single sugar, right? And of course, we actually learned 3 specific examples of monosaccharides. Glucose. What are the other 2? Anybody remember?
Galactose, fructose.
Excellent. There is a galactose as well as the fructose. Okay, so of course they can combine together to form different disaccharides. You can review that. As for the biological function, I'm just going to list a few here. Glucose would be the main source of energy production. Okay. Now we know it is in cellular respiration. Okay, so that's the glucose, main source of energy. In addition to being a source of energy, we can also store them as energy, right? So we have glycogen and starch. These are for energy storage. Glycogen is for animals. It's for us in the liver and the muscle. Starch is the storage form of energy in plants. They store it in the roots, in the stem. Okay? Like I'm not gonna write out everything, right? Like you can when you study, but you know, if I write out everything, we're totally gonna run out of time. And we have these things in the notes from the class as well, right? Okay, we can also use them for structural support, like cellulose, chitin. These are for structural support. Okay. Cellulose would be like the cell wall of a plant. Chitin would be exoskeleton of insects, crustacean, like the lobsters and the crabs and whatnot, right? Okay, I think that's pretty much all the stuff for carbohydrates. Okay, once again, it is CH and O. Protein is CHON plus a little bit of S. And the monomer of the protein is actually, anybody remember what the monomer of proteins are? What are
Polypeptides.
the polypeptide is the long chain, yes, but what are the individual building blocks?
Steroid.
Steroid is an example of lipids. We will get to that in
Amino
a
acid.
second.
Amino acids.
That's right, amino acid. Thank you. Amino acid, right? And there are 20 of them. Okay, I'm not going to ask you to draw anything on the test, but you should recognize what an amino acid look like, okay? You should recognize what an amino acid look like. I know it's a lot of stuff, but that's the pre-health program. This is the carboxyl group. And then over here, that's the amino group. Okay, so when you put two of these things close to each other, you can create a peptide bond, right? To connect them together. Okay, the carboxyl group, you can take away the OH. And then for the amino group, you can take away the H. That will allow you to take out the H2O, part of the dehydration synthesis, and you can create a peptide bond. Okay, so all 20 amino acids are going to have the same core structure. What makes them different from each other is the R group, which is the side chain. Okay, so there are 20 different side chains. Some are polar, some are non-polar, some are acidic, some are basic, some are positive, some are negative, right? So they behave differently. Okay, biological function. There are a bunch of them. So these are in no particular order. I'm just writing them as they come to my mind, okay? So energy source. Okay. Amino acids and be fed into different parts of cellular respiration. We had a slide on this in lecture 5, right? Some amino acid can be fed into glycolysis, some can go into pyruvate oxidation or Krebs cycle, and you can get some ATP from them. Okay. Protein is, of course, part of a balanced diet. We get our proteins from meat or soy products, and we burn them for energy. Proteins are also important for structure, right? Such as what kind of structure in the body is made up of protein?
Muscles
Mm-hmm.
and hair.
Yes, very good. Okay, so muscle is a lot of protein. That's why we also eat meat, right? Meat is muscle, right? To get proteins. Hair, right? Nails. Good. We also use proteins for enzymes. We just reviewed enzyme earlier. And an example of enzyme would be like lactase, right? Lactase. That's pretty much the only one we've learned. Okay, we'll learn more enzymes later on, maybe next semester. Okay, energy source structure, enzymes, hormones. And hormones are like chemical messengers, right? Not all hormones are proteins. Some hormones are lipids. Someone mentioned steroids earlier. That's also useful for forming hormones. The hormone that we learned for protein is insulin. Right? You probably heard of that for regulating blood sugar. That's a protein hormone. Okay. Now there are also a bunch of other stuff. Okay. For example, membrane proteins. Okay, membrane proteins. These are the proteins that are going to be stuck in the cell membrane, right? Some are integral protein that goes through. Some are like, peripheral proteins that are attached on the side. And they have a bunch of different functions, right? 6 of them, I believe. Just going to briefly list them here. Attachment to cytoskeleton. Do you guys remember what I'm talking about? Like this is beginning of lecture 4, right? What else are there? Attachment to cytoskeleton of the 6 functions. Give me one. Give me one more, please.
Also recognition.
Yes, cell recognition. Is that what you said? I think that's what I heard. Okay. Transport, right? And then there are also enzymatic activity, cell signaling, the Halloween story I told you about. And also, besides cell signaling, there's one more, tight junctions. Intercellular junction, I think that's what it was called. Intercellular junction. And you want to staple cells together so things not going to leak between them. So these are all functions of proteins. Okay. See lecture 4 for more detail. And so like I said, when we're studying, we try to connect everything together as much as possible so you can form these links where when you think of one thing, allows you to think of other things that you know. That is a powerful way of remembering things, I think. Okay, moving on. Lipids. Lipids, we have CHO and a bit of P. So there are no monomer here. Okay, but there are different components. Instead, there are components. Okay, so I'm just gonna list some of the familiar components. Glycerol. There are fatty acids, saturated, unsaturated. Carbon rings. I'm sorry, what was that? Okay, maybe just a mistake. Phosphate head, right? And then there are some functional groups. Now, I'm gonna come back to this, but in lecture 2, we roll it down, right? Lipids, and then we say, okay, so like triglyceride has 1 glycerol, 3 fatty acids. Steroid has the carbon rings with the functional groups, right? So you can go back to the notes to help refresh your memory. Okay. Now, in terms of the functions, we have the phospholipid. Phospholipids, it is the major component of cell membrane. Okay. We have triglyceride, which is useful for long-term energy storage. It cushions and insulate as well. But again, I'm just like keeping it simple here. You can add more details if you want. Steroids. They are part of the cell membrane. Okay, and separately, not related to this, they also form hormones. Okay, like estrogen. I think there's also wax that we talked about, which form water repelling surfaces. Okay, nucleic acid, CHO, NP, nucleotide. Okay. So for DNA, it is ACTG. And then for RNA, it's ACUG. Instead of thymine, we have the uracil, right? So DNA, it contains instructions to make proteins. RNA is basically like a copy of DNA. Okay. Also, part of the ribosome, if you remember. Okay. There are different kinds of DNA. And sorry, there are different kinds of RNA rather. And we haven't learned any of them yet. Okay. But one of them is going to be a copy of DNA. The other is going to be a part of the ribosome. Okay. So I'm just gonna let you catch up a little bit. And then I'm gonna go through the next question here. The next question. The numberings are kind of wrong when I copied over, but it doesn't matter. I'm sure you can follow it just fine. Hold on a second. For this one, I'm just gonna copy the answer for you so you can take a look, because we are not gonna have enough time to write out everything. How is the shape and structure of a protein stabilized? This is basically talking about the primary, secondary, tertiary, and quaternary structure. And we've done this a couple times before. So I'm just gonna copy it over for you. Please review that yourself. There it is. Okay, so the primary is like basically the sequence itself, and then secondary is the helices and beta sheets. Okay, so it's all here. You can review that yourself. Okay. Last question, which I also have to copy over. I wish there is an easy way to do this. Okay, identify the following molecules. Okay. I've Drew this for you a couple of times already. You should be able to recognize what it is. Anybody? What is this guy? Okay, we literally drew this a couple minutes ago.
First one is protein.
Okay, yes, it's part of a
From a
protein.
hydroxyl group and amino.
Yeah, amino.
Amino acid.
Yeah, excuse me, amino acid. Yeah, amino acid. Okay. This here, I see three things, and this is nucleotide. The phosphate is sugar and the nitrogenous base. This one happens to be a purine. Over here, this is a glucose. Okay, how about this? Has a bunch of C and H in it, very long.
Fatty acid. Fatty acid.
That's right. Do you think this is saturated or unsaturated?
Saturated.
And how do you know?
It's a single bond.
That's right. There are no double bonds, where everything is single, so it must be saturated. Very good, very good. Okay, that's it for lecture 2. Of course, you still have to do some more review on your own, but that's that for now. Okay, let's see. Sorry, I'm gonna have to skip the break, guys. Not gonna have enough time otherwise. We are okay. Anyway, like I said, the numbers are all wrong, but you can match it up yourself. List the major components of the cell membrane. We did this in glorious detail in lecture 3, right here. Okay, so I'm gonna ask you to refer to it. Okay, the fluid mosaic model of the cell membrane. There are, of course, the phospholipids, peripheral protein, integral protein, cholesterol, everything. Okay, so it's all listed here. Don't forget the glycoproteins and the glycolipids. They are responsible for cell recognition. That was just like our break time, so let's just ignore that. Okay, you have all these notes, right? I hope you do. Describe the central dogma of biology. Right? So the central theme, that's what dogma means. Central theme of biology is that we have information to make the proteins in DNA, and we cannot take the DNA out of the cell because it's very big and we don't want to damage it. So we make a copy of it in the form of RNA. Okay, so that process is called transcription. Transcription. Okay, hold on. Pen's not working properly. Okay, let me try that again. So that's called transcription. And transcription happens in the nucleus. So once you make a copy of the DNA, it's going to exit the nucleus through the pores, it's going to go into the cytoplasm, and the ribosome will come and use the copy to make protein. This is called translation. And that happens in the cytoplasm. Okay. One of the functions of liver is to remove toxin from blood. Based on its function, which two organoids would you expect to be abundant in liver cells? So detoxification, that would be things like smooth ER, peroxisome, okay, and even a little bit of lysosome as well. Okay, I know it's asking for 2. These are the main ones. Bit of lysosome as well. Okay, next question. Describe the structure of the mitochondria and state the function of each part. Okay, so since we learned that from lecture 3, we've actually learned a lot more details about the mitochondria, especially in lecture 5. So let's just try to review both of those things at the same time. Here is our mitochondria. That's the outer membrane. And then the inner membrane is going to fold up into these cristae. Okay, so the cristae is folded up to maximize surface area, right? And the innermost portion is called the matrix. So by now, you should know what happens at each of these locations. Oh, I forgot the intermembrane space. Okay, so tell me, guys, which process or which processes take place in the matrix? Anyone at all? There are 2 things, 2 parts of the cellular respiration that happens in the matrix. What do we? Yeah, Fahima, go ahead.
Krebs.
Yeah, Krebs cycle. Good. And
Pyruvate.
that's right. Pyruvate oxidation. Okay. And in the cristae, we're gonna have these electron transport chain, right? Which is just a series of proteins with increasing attraction to electrons. And these are ETC, electron transport chain. And the theme here is that the electron transport chain is going to transport the electrons, but more importantly, at the same time as it is transporting the electron, it's going to harness the energy from the electron so that it can pump H plus into the intermembrane space, right? So over time, we're gonna have a lot of H plus in the intermembrane space. And all these H plus wants to go back into the matrix because things naturally want to go from high concentration to low concentration. But it cannot do that on its own because the inner membrane is blocking them. So the only way to do so is to come back through the turbine, right? These are green little knobs. So as they go through the turbine, ADP will be converted into ATP. And so that's how you generate ATP. If you're wondering the name of the turbine, we also wrote that down last time. It's called ATPase or sometimes ATP synthase. Okay, so that's a really quick summary of the different things that are going on in the mitochondria. Not all the cells will have the same number of mitochondria. It depends on its energy requirement. Things like muscle cells, brain cells, they would have more mitochondria compared to something like a fat cell, okay, or skin cell. That's that. Are we okay? All right, last question here in the review for lecture 3. Describe the endomembrane system. So again, I'm just gonna quickly give you a review. It's probably better for you to just listen and absorb what you can, and then you can try to draw this out again later on your own or watch the recordings, okay? So this is the nucleus. And we talked about how the nucleus is surrounded by a membrane. You already have this note, in fact, okay? Like I'm just redrawing it to help you remember it. Okay, so that's the nuclear envelope. Okay, so the nuclear envelope does not end with the nucleus. It actually extend outward to form a network of these tunnels, right? And these ones have ribosomes stuck on them. Okay. So these are you label it on the quiz, so you should know that they are rough endoplasmic reticulum. Okay. These are part of the endomembrane system. They are membranes within the cell. So the rough ER, to put it simply, it will work with ribosomes, oh my goodness, work with ribosomes to synthesize and package proteins into vesicles. Okay. Now again, you don't have to copy this. You can copy it later. Okay, just try to listen, right? So coming out of the ER here, we're gonna have these vesicles. Okay, these vesicles with proteins in them. Okay, vesicles. Where do they go? What organelle do they go to next? Tell me, please.
Smooth endoplasmic reticulum.
Sorry, where did they go? I didn't quite catch that.
Smooth endoplasmic reticulum.
Oh, the smooth ER. Good try, not quite. Okay, they don't go to the smooth ER. They go.
Golgi body.
Yes, that's it. Okay, Golgi body. If you don't get it the first time, try again. You eventually get the right combination. So there is the Golgi body. Okay, the Golgi body is going to receive these vesicles. And what is it going to do with the proteins inside? Well, it's going to sort them, modify, and repackage proteins arriving in vesicles, right? So these things are going to go here. They're going to move through the Golgi body. And in the end, you will repackage them into a new vesicle. Okay. And then this thing can do different things. And you can look back at the lecture 4 notes. See, is it lecture note? No, lecture 3. See lecture 3 class notes. Okay, we roll it down. It can be sent out of the cell through exocytosis. It can become part of the cell membrane. You can store it in the cytoplasm. Okay? But since we're talking about the endomembrane system, it can also become a lysosome. Okay, so a lysosome is also part of the endomembrane system. They contains mixture of enzymes. Okay, so they can break down macromolecules, old organelles, and they could also trigger cell death. Right? We have a name for it, apoptosis. Okay? If the cell is not functioning properly, if it's too old, then the lysosome can release all of its enzymes and kill it. Okay. Also part of the endomembrane system, and we didn't draw it yet, would be like over here. Right? There's going to be like more membranes, but these ones don't have the ribosomes on them, so they are smooth ER. They are for lipid synthesis or breakdown, also detoxification. Okay, so there you have it. That's the endomembrane system. I'm just gonna type a quick note here. There are other organelles that you have to study. It's not on this sheet. There's the centriole, right, for chromosome movements during division. The cell wall, central vacuole, a contractile vacuole, peroxisome. Oh yeah, well, ribosome as well, which makes protein. Cilia, flagellum, cytoskeleton. Okay, so these are some additional ones. Oops, mitochondria we did already. Chloroplast for photosynthesis. I think those are the key ones, okay? In addition to these guys over here. All right, that's lecture 4. Anything you want to ask before we move on? Anything at all? Just let me know. No? Everybody good? Already. Next, we have membrane transport. Okay. Explain what passive transport is. Provide example of passive transport. Describe the difference between various type of bulk transport. Basically, for lecture 4, you have to study this flow chart. Okay, we wrote this down. Like basically all the important key stuff are in the class notes for lecture 4. All right, like please go through the isotonic, hypertonic, and hypotonic stuff yourself. And then there's this flow chart that will summarize all the different kinds of transport for you. Okay. I'm not gonna go through them again, but I will copy it and paste it over here for you. Okay. Let's see. Okay, so this question, what passive transport is? Passive transport is over here. It's from high to low concentration, no ATP needed. This is what you should know. Okay, you should know examples of each type of transport. Okay. For example, if I ask you, if I just, oops, what is going on here? On the test, I could just give you something. Hold on, what is this? Oh, this is wrong. Okay, on the test, I can just give you like an example. I'll be like, facilitated diffusion. Okay, and you should know, oh, that's part of passive transport. I can list a bunch of transport mechanism, facilitated diffusion, osmosis, exocytosis, phagocytosis, list all of them, and I ask you which of these require ATP. What I'm really asking you is which of them is part of active transport, because active transport needs ATP, right? So memorize it if you can. If not, have this chart next to you, okay, as reference. Then you can quickly check to see which one is what, right? So you should know each type of transport, know the differences and similarities between different types of transport. For example, even though facilitated diffusion is passive and protein pump is active, so that's different. One is from high to low, the other is low to high. One requires energy, the other does not. But how are they similar? Well, they are similar in the sense that they both involve some kind of membrane protein, right? Okay, so make sure you know this chart, know it well, basically. Okay, moving on. Oh yeah, this one. Okay, this one was on the previous quiz. And let me just copy it over first. The cell of a plant that grows in very cold environment was studied. What kind of adaptation do you expect the cell membrane to have? On the quiz, you get one of these. You got one of these as well, right? Like I forgot. Like some of you got the hot environment, some of you got a cold environment, depending which quiz you wrote. But basically, it was 4 marks, and this was all I was looking for. Okay, adaptation. Okay, so if it's a cold environment, the adaptation is it needs to be more fluid so that it's not going to freeze up. So for it to have more, for it to be more fluid, you need to have more cholesterol, more unsaturated fatty acid, and less saturated fatty acid. Okay, so this is for cold. That's the 4 marks. 1, 2, 3, 4, right? Okay. And then for hot adaptation, okay, which I know it's not what this question is asking, but in case it would be less fluid, and then it would be the opposite. Okay, you need less cholesterol, less unsaturated fat, and more saturated fatty acid. All right, that's that. Okay, number 5 now. Just copy it over. What is an organic molecule? An organic molecule is basically contains carbon atoms, right? Except for CO, CO2, and CN. Even though these contain carbon atoms, but the people who came up with the definition like, oh, let's not count them. Okay, next, list the products of glycolysis. Actually, we had a nice chart from before, which is right here. So not just for glycolysis, but for all 4 stages, right? So I'm gonna copy this over for you. There we go. Okay, so the main products of glycolysis would be pyruvate, because you split the glucose in half, right? You generate 2 net ATP and some energy carrier. Okay, so make sure you know what are the key things that you created. Okay. In cellular respiration, glucose is oxidized while oxygen is reduced. Explain what it means. Well, glucose is oxidized, meaning it's going to be, there's a loss of electrons. And then it becomes carbon dioxide ultimately. Whereas for oxygen, it is reduced, meaning it's going to gain of electron. And then it's going to become water. Okay. But it's important to remember this transfer of electron is not a direct transfer, right? After our explanation of everything that goes on in cellular respiration, you should know that, we started with glucose, okay, and ultimately it becomes carbon dioxide. Okay, but along the way, I can't change color all of a sudden, but doesn't matter. Along the way, we're gonna be releasing electrons. Okay, so that's why this is called oxidation. Okay. All these electrons are captured by NAD plus and some FAD. Okay. And then they become NADH and FADH2. And these things are going to go to the ETC. Oh my goodness. Let's just try to restart one more time. Okay, so these energy carriers, they're going to go to the ETC, right? The purple stuff, right? And then they're going to drop the electron off, and then electron is going to bounce off the ETC. And at the very end, you have the oxygen. Oxygen is called the final electron acceptor. I remember the story about getting the kid off at the bottom of the slide. That's what the oxygen does. It gets the electron off the slide, and when it does, it becomes water. Okay, so the electrons originates from glucose, but it has to go through all these steps in between before it ends up on oxygen, right? This is the redox reaction that we refer to when we discuss the cellular respiration. Okay, what is the role of NADH and FADH2? So as I described to you here, they carry high energy electrons to the ETC for ATP production. Okay, and just to remind you, this method of producing ATP at the ETC, it's called oxidative phosphorylation. It's the common way of making ATP, right? 90% of the ATPs are created this way. What is the significance of adding coenzyme A to pyruvate during pyruvate oxidation? Okay, so this happens in stage 2. By the time we get to stage 2, much of our investment, the 2 initial ATP that we invested, much of that energy has been used up. So the purpose of coenzyme A is basically to re-energize the molecule for further reactions. Okay. All right, number 7 here. This kind of calculation question is definitely going to be on the test. Can you guys tell me what the answer is? Do you guys remember what to multiply by and what to add? What's the answer? How many ATPs can each NADH produce?
3.
3, that's right. So we're going to take 50 times 3, which gives me 150. What about FAD?
2 ATPs.
That's right, and that's 38. And when you add them up, is 188. Okay? 188 ATP. We also did one in class. I don't know if you remember. It's like, okay, this is not related to this question, okay, but like, 7 cycles of 7 spin, I think this was called 7 spins of Krebs cycle. Okay. So you have to remember one cycle. It's in the chart, right? So one cycle, we make 1 ATP. This is direct production through the enzyme. 3 FADH. 1, oh, sorry, 3 NADH, 3 NADH. 1 FADH2. Right? This is per spin, not per glucose. Right? If we talk about per glucose, then each glucose spin twice, right? But this is one cycle. Okay? One spin. One spin. This is going to times 3 times 2. This is just 1 ATP. If you add them up, it's 12. Okay? So you can remember one Krebs cycle spin is 12 ATP. So if you do it for 7, okay, 12 times
84.
7. Yeah, thank you. Okay. That's it. Okay, the inner mitochondrial membrane area is about 5 times. Okay. So why is that? Well, before we say the inner membrane forms crystate, right? More area equals more ETC equals more ATP production. Okay. You can pack more of those proteins in it. You can pump more H plus. And then you can create more ATP in return. Which step of cellular respiration generates the greatest number of ATP? That would be the ETC. Okay, 32 ATP. 32 ATP. 2 directly from glycolysis, 2 more from Krebs cycle. Most of it comes from the ETC. Describe what happens as H plus is pumped from matrix to inner membrane space. Okay. Remember, it's like the dam I told you about, right? Okay. This is like the inner membrane. And then all this water that you build up, that's basically the H plus. Okay. Where does the H plus want to go? They got to go back down here, right? Okay. So that's kind of like, this is the matrix down here. Up here is the intermembrane space. Okay, so the inner membrane is in the way, the dam is in the way, right? This is like the dam. It's not going to be able to come back down. We can dig a tunnel for the water to go through. Okay. And we can put a turbine, right, which is the ATPase, right? ATPase. That's like the turbine, right? So as the H plus goes back in, it's going to turn the turbine, and that will give us the ATP. Okay? So it's all about creating that H plus reservoir in the intermembrane space so that when they come back in through the ATPase, it will generate ATP for us. What is the purpose of the final electron acceptor? Anybody remember what the final electron acceptor is? We wrote down a little bit ago, but just wondering. What's the final?
Oxygen.
That's right, this is the oxygen. Okay. The purpose of oxygen is to remove electron from ETC so that the electron flow can continue. And remember, if the electrons are not floating down the ETC, we are not creating the H plus reservoir. Without that, we cannot create ATP. Okay. So that's that. This is all the stuff that is on the review sheet. There is a review quiz that you can do, review tests, practice tests, whatever you want to call it. The link wasn't working for some people. I just got an email before the lesson. It should be fixed now, but for some reason, if it still doesn't work, just email me again. Okay, I'll let you know. So all the notes and answer keys are updated. This is the thing we just took up. This is the link to the actual test, which will happen this Thursday. No lecture that day. 100-minute, 44 multiple choice, 10 multi-select questions. So that's like 54 questions, 100 minutes. So that's around like 2 minutes per question. Should be enough. There is the practice quiz that I talked about. Okay, and this is all the poll question answer. You know those concept track questions that we do in class? We do the Zoom poll, right? You can download a copy of all of them plus the answer as well. This is a very good animation for cellular respiration. If you haven't watched it, please do so. And that is it. Okay. Now, no class next week because it's reading week, engagement week. You get a bit of a break. But when we return, though, okay, the second half of the course, just a little warning, it is even more difficult than the first half. So please continue to come to class, and good luck with the test. As usual, just email me if you have any question. Okay? Thank you, guys. Take it easy. Bye for now.